Some species of tardigrades, or water bears as the tiny aquatic creatures are also known, can survive in different environments often hostile or even fatal to most forms of life. One could easily imagine that by decoding their secrets, we could apply the knowledge to ourselves to make humans more resilient to extreme temperatures, pressures, and even dehydration. This is just science fiction for now, but nevertheless, researchers, also captivated by the microscopic creatures, seek to understand the mechanisms responsible for their robustness, as this could bring other benefits too. Although water is essential to all life we know of, some tardigrades can live without it potentially for decades. The trick is in how their cells deal with this stress during the process of dehydration, said Associate Professor Takakazu Kunita from the University of Tokyo's Department of Biological Sciences. It's thought that as water leaves a cell, some kind of protein must help the cell maintain physical strength to avoid collapsing in on itself. After testing several different kinds, we have found that cytoplasmic abundant heat-soluble CAS proteins, unique to tardigrades, are responsible for protecting their cells against dehydration. Recent research into CAS proteins reveals that they can sense when the cell encapsulating them become. The process is reversible, so as the tardigrade cells become rehydrated, the filaments recede at a rate that doesn't cause undue stress on the cell. Interestingly though, the proteins exhibited the same kind of action even when isolated from tardigrade cells. For one thing, in order to visualize the proteins, we needed to stain them so they show up under our microscopes. So we turned to a methanol-based solution to get around this problem. Research on mechanisms related to dry preservation of cells or organisms could have many future applications. This could extend the shelf life of materials used for research, medicines with short expiry dates, or maybe even whole organs needed for transplants. For a biologist, this field is a goldmine, said Kunita. I'll never forget New Year's Day 2019, when I received an email from Tomomi Nakano, another author of the paper. She had been working late trying to see the condensation of CAS proteins and observed the first CAS filament networks in human cultured cells. I was astonished at seeing such clearly defined microscopic images of these. It was the first time I had seen such a thing. It was a very happy new year indeed. Knowing how to isolate and activate these special proteins, though, is just the beginning. University of Tokyo. 2022, September 6. How tardigrades bear dehydration. A new mechanism explains how water bears survive in some extreme conditions. https://ift. tt. 3-ear1 pcd. Access September 6, 2022. Thanks for watching. Please subscribe my channel for more videos.